Hi everyone, welcome to class. Today we're going to start our first drug. How exciting! I bet you're all so excited. So our first drug or ligand or neurotransmitter, whatever you want to call it, is acetylcholine. Now, before we jump straight in, we have to go over what is a neurotransmitter, because acetylcholine is a neurotransmitter. Turns out there's criteria for what makes up a neurotransmitter. From here on out, it'll just be NT. So there's 6 criteria that we need to follow. First is that the presynaptic cell should contain this substance as well as some sort of mechanism to synthesize it. So with acetylcholine, that means it's synthesized in the presynaptic cell and stored there until it's ready. The substance should then be released when depolarizing stimulus is applied to the neuron. There is then going to be some sort of receptor on the postsynaptic cell for our neurotransmitter to bind to. There should also be some sort of known antagonist to block the effects at this receptor. There should be a system to inactivate the substance. So, in other words, to remove the substance from that receptor. Okay, so neurotransmitters must be able to be removed from receptors. There should also be exogenous application of the substance to the postsynaptic cell should produce the same response as if it was normally produced or normally released and acted on that synapse, that receptor. Okay, so if we were to take acetylcholine and inject it directly into the synapse, so we cut out that presynaptic cell, it should cause the same effects as if the presynaptic cell released it itself. Okay, those are our criteria for a neurotransmitter. So the majority of what we're going to talk about for Unit 1 and some of Unit 2 of this class is going to be covering different neurotransmitters and how they work at the synapse and what kind of effects they have. We also have things called neuromodulators. So these are things that a lot of times will be like hormone-like. So they tend to act at a distance rather than on that synapse directly. Okay. So there's different types. We've got autocrine, juxtacrine, paracrine, endocrine, exocrine. Okay, so the pre-word tells you where it's acting, and then crine means at a distance. So autocrine, self-acting, but at a place other than the synapse. Okay, endocrine, far away from this. But again, acting on something other than the synapse. So, a neuromodulator does not elicit direct effects on that postsynaptic cell, but it will alter the actions of a classic neurotransmitter. Okay, so it could enhance the neurotransmitter actions, it could reduce it, or prolong it. So sometimes these neuromodulators will actually act on the presynaptic cell, again, at someplace outside that synapse. It could be something that acts on an astrocyte, or it could act on the postsynaptic cell, but indirectly causing changes to the effects of the neurotransmitters. Okay? So again, it may function either anterograde or retrograde manner, whether it's pre to post or post to pre, and it may be released from glial cells. So glial cells like astrocytes. Those are going to be the most common glial cells we'll talk about in this class. Okay, let's get into acetylcholine specifically. So we're starting with acetylcholine because it turns out it was actually the first neurotransmitter identified. So if you like history, I like history. It was actually a German pharmacologist in the 1920s that performed an experiment on frog hearts. And it was during this that he developed or kind of identified acetylcholine. So pretty much there were two hearts. They were live dissected. One of them had the vagus nerve intact. And when we bathed those hearts in a buffering solution, then the first heart was stimulated electrically, so via that vagus nerve, and it slowed down the heartbeat. After a period, the media was collected and applied to the second heart, and it also slowed it down. So that first heart that was electrically stimulated released a fluid or something, and when that was applied to that second heart, it caused the second heart to slow down as well. Now, this is the experiment that kind of figured out that something's being released, and that's where we got neurotransmitter, but it did take several years for that chemical to be identified, and it was later identified as acetylcholine. So that's the history. I just think it's really cool. Also, who thinks to do that? You know, it's weird, but cool. So acetylcholine was first used in these experiments to determine the nature of a chemical transaction at the synapse. So it's very common at neuromuscular junctions in the periphery. Okay, so where neurons connect to muscles, causing muscles to move, to contract, or retract. That's the opposite, right? So any neuron that uses acetylcholine, we call a cholinergic neuron. Okay, so cholinergic means using acetylcholine. So you will hear that term over and over. So going on, this is the synthesis of acetylcholine. Yes, you do need to know this. You need to know that it's acetyl coenzyme A. You need to know choline. You need to know chat. You need to then know acetylcholine. Okay, for every neurotransmitter and drug we're going to talk about, you do need to know the synthesis and metabolism of these drugs. Okay, you don't need to know the chemical structure, but you need to know the names. So, chat is choline acetyltransferase. Okay, so it's a type of enzyme that helps in the synthesis. And it is used to identify cholinergic neurons in the central nervous system, because only cholinergic neurons have this synthetic machinery for producing acetylcholine. And we can visualize chat by using immunohistochemistry. So we make antibodies that target chat itself, and then boom, any neuron that lights up afterwards we know contains chat and therefore is a cholinergic neuron. Because neurons that do not contain chat cannot create acetylcholine. So synthesis, acetylcoenzyme A plus choline using chat equals acetylcholine. Okay. Now, I mentioned that we can use chat to identify these neurons. This is an example of what we would see after that immunohistochemistry. So this is just showing the cholinergic motor neurons in the ventral horn of the spinal cord. So anything that's here, you can see it's green. Those are neurons that contain chat. So then you can see here, this would be a muscle probably, or other types of neurons that do not contain chat. So therefore, these are not cholinergic cells, but these are cholinergic cells. Okay. Chat is also only expressed in the cytoplasm of the neurons. So we see it mostly in the soma, but a little bit in the axons. And that's just because that is where acetylcholine is synthesized. So how do we get acetylcholine into our systems? You know, we have to have acetyl coenzyme A, but we also need to have choline. And it turns out we can get choline from our diet. And that's actually where we get the most of our choline in our systems, is from our diet. And we get it from the hydrolysis of dietary lipids. And choline is water soluble. So it's capable of crossing the blood-brain barrier. Obviously, that means it has to be cleaved from these lipids, because the lipids are not water-soluble. So we have these dietary lipids. The choline is then cleaved off of it, and the choline can then cross that blood-brain barrier using specific choline transporters. Okay. So a lot of foods will contain choline, especially those foods such as eggs, legumes, red meat. So things that have tend to have a lot of protein also tend to have choline. So red meat, organ meat, so especially liver. Liver actually has a lot of choline in it. If you're not eating enough of these foods, you may have a deficit of choline in your system, and that can bring on some health concerns, especially in like endurance athletes and alcoholics. They might need more choline than others, so they just need to make sure that they're eating enough of these foods that contain it. Deficiency in this could lead to memory loss, but we're not quite sure. It could also be playing a role in cognition and attention. Okay, so we have acetylcholine. Let's say it does its job in our system, and then we need to break it down. So acetylcholine is metabolized back into choline and then into acetate. So either acetic acid or acetate. And it's done by this enzyme. It's called acetylcholinesterase, okay, ACHE. So in contrast to chat, ACHE is expressed by multiple different number of cells, including muscle fibers. Okay, so non-cholinergic cells can contain acetylcholinesterase. Okay, only cholinergic neurons have chat. Other neurons can also have acetylcholinesterase. Now, so that means that marking this is not useful for identifying cholinergic neurons. Okay, so that's why we use chat. So again, yes, you need to know the metabolism of acetylcholine. What breaks it down and what are those products? Okay, so here is just a fun diagram from your textbook showing what we just talked about. Here, purple, presynaptic cell, blue, postsynaptic cell. In that presynaptic cell, we have choline, acetyl-CoA, and then we have chat acting on that, creating the synthesis of acetylcholine. Acetylcholine will then be moved into vesicles, and then it'll be put or released into the synapse when there's the right charge, right? Very typical synapse, but you see that the neurotransmitter is synthesized in that presynaptic cell. There will then be receptors for acetylcholine on that postsynaptic neuron, okay? You'll see here that there's multiple different types of receptors that it has, and we will talk about all those. I believe we're going to talk about all of those in the next class. So for now, just know that they're all there. Acetylcholine is then going to be broken down, and it's actually broken down in the synapse, okay? So metabolism occurs in the synapse. So then choline and acetate will then be taken back up by that presynaptic cell, and they can then be recycled and used to create more acetylcholine. So the cholinergic systems have the choline anabolic enzyme, or CHAT, of course, in that presynaptic, and then the catabolic enzyme, ACHE, is in the synapse itself, okay? And this is what's going to differ across different types of neurotransmitters, is where does that metabolism, that breakdown occur. That's going to change on the neurotransmitter. So make sure you remember where it's occurring. Okay, let's stop this video here, and in the next video, we'll talk about how acetylcholines are used.